Good morning, good afternoon, and good evening, everyone. Thank you very much for joining us today. I'm Stoyan Stoichev from the Resin Biosciences team, and I'll be the host for your session today. Before we get going with the webinar, just a couple of housekeeping announcements. We are recording the session, and we'll make this available in the next couple of days via social media channels. You can post your questions using the Q&A box at the bottom of your screen. We'll have plenty of time to address this after the presentation. So today is the first industry-flavored episode of Emerge. So here we're focusing on proteomics technologies that have made or are making the transition from academic to a more routine environments, as well as how these technologies and methodologies are having an impact within their respective industries. So today, our speaker is Dr. Marco Tognetti. He's a principal scientist from the diagnosis team, and the bulk of his time is spent developing novel sample preparation solutions. Marco completed his PhD as well as the remainder of his postgraduate studies at ETH Zurich, and the bulk of his work was focusing on applying multi-omics tools to study the drug responses in lung cancer. And today, he'll be presenting some of the work that he and his team have put into developing and implementing a high throughput and sensitive workflow for immunopeptidome profiling from low input amounts, as well as how this workflow is being applied within the precision oncology arena. So Marco, thank you very much for joining us and coming on board for this platform. And I look forward to your presentation.
Thank you very much, Stoyan, for this opportunity. And also I am really much looking forward to presenting this work with you guys and discussing that. As a start, I would like to thank the people that actually did a lot of this work, with a special thank going out to Luca and Ilya. And today's topic, I want to touch on 5 points mostly. The first one is why, what are immunopeptides? The second one is why should we care at all about them? The third one is how is the quality of the immunopeptidomics data that we generate? And then in the last two, with two practical examples about two biological cohorts that we analyze, if we can find biological information. And in the last one, if we can find neoantigens. The mammalian immune system is composed of a layered defense system, and it has increasing specificity. The innate immunity has physical, chemical, and cellular barriers that are non-specific. So they tend to be a very broad specificity and tend to stop pathogens. However, if this is not effective, the adaptive immunity kicks in, and this is highly specialized and generates an antigen-specific response that is tailored to the specific pathogen that it wants to defeat. How is antigen specificity ensured in humans? It is mostly through the non-self recognition by T cells. And this happens through the T cell receptors that then bind to MHC-associated peptides or immunopeptides. So here we have the T cell receptor in green, and at the bottom we have here in red the MHC or HLA in humans, that it's bound to a MHC-associated peptide or this immunopeptide, and this is the antigen that is then recognized by the T cell receptor. Interestingly, the HLA is encoded by the most polymorphic gene cluster in the human genome. So more than 17,000 allelic forms have been reported to date. And interestingly, these kind of allelic forms or allotypes alter the structure and specificity of the peptide binding cleft, and thereby defining a different subpopulation of peptides that can be presented and recognized by T cell receptors. An important thing to know is that the fact that if a peptide is presented, it does not mean that it will induce an immune response, because there is still this non-self recognition that it's based on the T cell receptor recognition. And it's known that both qualitative and quantitative changes associated with the immunopeptidome are associated with disease. And there are two classes of MHC or HLA molecules in humans, and I will now walk you through these two different classes and why we have two. Class 1, it's presented on any nucleated cells. Each cell can have up to 6 different allotypes presented, and they are present in high abundance. The peptides bound by HLA class 1 are between 8 and 11 amino acids in length and are very constricted because the binding cleft is very defined. And it presents these antigens to cytotoxic T cells, and if these cytotoxic T cells recognize this as a foreign antigen, it will elicit cell death. So it is a key player in cell-mediated response. On the other hand, class 2, it's presented only in immune cells. Each immune cell can present up to 16 different allotypes, so it's a very large variety and different peptides that can be presented. These peptides are also much longer, between 10 and 25 amino acids in length, and are also more variant because the binding pocket is open on both sides, so it can fluctuate a bit the length. And it presents the antigens to T helper cells, and it has thereby a key role in humoral response. So what are immunopeptides? They are the key player of the antigen-specific adaptive immune response. So the application fields of immunopeptidomics, it's very broad. It can be any disease susceptibility because it's linked to any disease. It's very interesting when it is too active, and this is the case of autoimmune diseases or graft versus host diseases. It is a hot topic in the personalized cancer immunotherapies field, where we can distinguish between neoantigens that come from tumor-specific mutations and tumor-associated antigens that come from tumor-specific overexpression. So you find these antigens on healthy tissues as well, but they are specifically overexpressed in tumors. And this is, of course, also very a nice field for the design of novel treatment methods, whether you are thinking about vaccines or cell-based therapies, knowing what is actually presented and can be targeted, it's of course very nice. So why should we care about immunopeptidomics? As they are the key of the specific immune response, its understanding has the potential to lock, in the case of autoimmune diseases, we would want to lock it, or unlock it in the case of cancer, because this machine is very powerful. Why it is not more widespread? Because it comes with a lot of challenges. The foremost is that it usually requires a very large sample input in the grams range. The peptides have limited physicochemical variability because they are bound specifically. They often ionize poorly because they don't have the basic amino acids that tryptic peptides usually have. This also leads to non-conventional fragmentation, so the Y series of ions is not as highlighted as you would expect. And the search space is, of course, huge because you don't have the digestion specificity. And this becomes even more of a challenge when you are looking for neoantigens. And especially these last 2 points, it's why the field has been developing so much in the last years. It's just because the search algorithms have become that good that can deal with that. So I would like to walk you through our workflow that enriches for immunopeptides. What is important to know is the fact that there is no digestion needed, and that by subsequent enrichment of class 1 and class 2, we can generate 2 samples from one tissue or cell. So we start with cells or tissues, it doesn't matter. We perform a native lysis, and then under native conditions, we perform a magnetic immunoprecipitation, where we have magnetic beads bound to antibodies that recognize specifically either class 1 or class 2 HLA molecules that are in turn bound to their immunopeptides, here highlighted by these lines here. Then we perform a high throughput peptide separation because we want to separate the peptides from the HLA molecules. We measure it with our true discovery pipeline, that it's LCMS-MS, and then analyze the data with our in-house software that are SpectroMine and SpectroNaut, which we had also to improve significantly in order to support this kind of pipeline because of the challenges we discussed earlier. So we performed extensive optimization of this pipeline. We did 33 rounds and measured more than 1,000 samples. So we know exactly how this pipeline works, and we optimize it extensively. Now I would like to share with you some of the key steps that we found during these optimizations. First of all, the lysis is very important because we want the environment to stay native. We don't want any denaturation to occur. And we found adapted focus acoustics to work really well because it's highly controllable. Then, given also this platform, one thing that we really found very good is the possibility of doing it in a semi-automated manner. And this is possible only because of the usage of magnetic beads. And here we use protein A linked magnetic beads, and this really allows the processing of 96 well plates, and you don't have any clogging or any of those other kind of problems. Another very important part is that we introduced the FAMES, so the ion mobility based separation, to clean the spectra, and this helped a lot as well. And of course, being a mass spec company, we also put a lot of effort in generating very good DDA and DIA methods for these specific kind of samples. Of course, every time you have a pipeline, one of the first things is how good is the quality of the data that this pipeline, so to say, spits out. To assess that, we process 20 million cells in triplicates of JY cells. This is a classical immune cell line. And the first question is, of course, how reproducible are measurements? By the use of DIA, we could reach a close to 90% reproducibility. So with more than 10,000 modified peptides found in all 3 of the replicates. And this is the data for class 1. We repeated that subsequently for class 2 as well. And here you can see also the fact that in class 2, the reproducibility is a bit less. This is likely due to the fact that it's the second immunoprecipitation. So it's the second step. So every time you introduce a step, you introduce also variability. But overall, almost 10,000 peptides are found every time you measure this sample. So the reproducibility is very high. What you can also look at, and we discussed in the introduction, is the fact that peptides have very clear physicochemical properties due to the fact that they are bound by these specific HLA molecules. For the class 1, we know to expect peptides that are between 8 and 11 amino acids in length and have a very sharp peak at 9. And this is indeed what we found. For class 2, this is a bit different. As we discussed, the peptide length is a bit broader, and indeed we find peptides that are mostly 15 mers, but the peak is also a bit broader than for the case of class 1. Another possible QC measure is the anchor positions. We discussed the fact that different allotypes bind preferentially specific peptides. And what we know is that for class 1, again, since the peptide binding cleft is so defined, these positions are conserved. So we have position 1 and position 8 of the peptide that are, so to say, fixed. And there is a very strong preference for specific amino acid to bind on those positions. So what we can do is, for our data set, look at the fraction of peptides that have specific amino acid in position 1, for example. And here, representing the triplicates in different colors, again, seeing the high reproducibility of the pipeline, and we see a very clear preference for proline, leucine, and valine. And we can then go in the literature, since this JY cell line, we know exactly which allotype it's expressing, we can then check which kind of preference for position 1 is actually known. And we can highlight that in green, and we saw that we recapitulate the expected phenotype. We can repeat the same for position 8. We see that the preferences here are different. This is also known, but then if you look in the literature, this is also recapitulated. Another thing that you can do once you know the allotype, you have different prediction algorithms out there that predict how strong different peptides bind to the different allotypes. And if we run our data through such a pipeline, we find that more than 80% of the peptides are predicted as strong binders. Again, giving us confidence that we are very good at enriching for this kind of peptides. So to summarize the quality of the data, the reproducibility is really good. the peptide length distribution matches published data sets. And if we have the allotype information, and this works better for class 1 than for class 2, we can also check the anchor positions and the predicted binding affinity. And overall, the quality of the data is comparable to published data set in all these different metrics. Very nice. Next step was, we know that usually this pipeline needs a lot of tissue or a lot of starting material. So our question was how much fresh frozen tissue is actually needed to reach a plateau or reach very good numbers. So we used healthy lung tissue and perform a tissue ramp. So we started with 2.5 milligrams and went up to 135 milligrams. And then on the y-axis, we plot the number of modified peptides that we could identify. And for class 1, the picture is very clear. So with already 10 milligrams, we get a comprehensive picture of the class 1 immunopeptidome, and we get more than 10,000 modified peptides. We repeated this for class 2. And here the picture is a bit different. So we see a steady increase on the number of modified peptides we could identify. And this is likely due to the fact that the class 2 immunopeptidome is slightly more complex, as well as more heterogeneous, because you have different populations of cells and immune cells that can express them. What we can overlay is the usual biopsy size. Here, I will portray that in red. And we see that we can already, with 15 milligrams, it's something that you can get from a needle biopsy, we would get already more than 10,000 peptides from one single sample. So it is a more sensitive pipeline that published workflows to date. Nice. So next we asked, can we actually apply it in a more biologically sensible cohort? So we used a matched lung cancer cohort where we had 12 patients with matched lung tissue from a healthy region and cancerous region. We had 15 milligram each, and we processed that for class 1 and class 2 immunopeptidomics. Overall, we could find more than 100,000 peptides with an average of about 10,000 per sample. So it's a very sensitive enrichment from as little as 15 milligram tissue. What we can also plot is for the different samples that we have matched. So we have here, for example, for patient 1, cancer and healthy, we can then plot the number of modified peptides for both class 1, that will be in gray, and class 2 in red. And what we saw is that there is no significant difference, for example, between cancer and healthy tissue. And overall, class 1 tend to be less variable in the numbers than class 2. We also processed the entire cohort twice. Once we measure with our optimized EDA method and once with the DIA method. And this is just the average between the two different methods. And we found, as already published, that DIA is much more sensitive. And this has very well-known reasons. This is for class 2. For class 2, we have an even higher increase in sensitivity. And this is likely due to the fact that class 2, as we saw also for tissue ramp, is a more complex immunopeptidome. Next, we asked, how is this in class 2 immunopeptidome of the matched tissues? To address that question, we started very simple, just a hierarchical clustering of all the modified peptides that we could identify in the different samples. And the picture we got was, so to say, a bit messy. So there was no clustering based on disease or individual. However, there was clustering based on the GO term to which the proteins, the peptides of which were presented happens. For example, here at the bottom, we have these two samples that have an increased presentation of peptides that map to proteins that respond to epidermal growth factor. But overall, the class 2 immunopeptide presentation is very variable. And this is also possibly due to the fact that tissue heterogeneity, again, going back to the tissue ramp experiment. And likely, maybe, if you would process more of the tissue, you maybe would get better coverage. We repeated the same for the class 2 immunopeptidome, and we found a very striking picture. So here, the samples cluster perfectly by patient. Doesn't matter if cancer or healthy tissue, they cluster together. So the cancerous tissue does not dramatically change peptide presentation on HLA class 1. This also makes, again, sense given the fact that these HLA class 1 are presented in all nucleated cells. And so also you don't expect super strong differences between cancer and healthy tissue at the protein expression level. However, if you do and go and look at differentially abundant peptides between healthy and cancer tissue, we could find a lot of immunopeptides that are significantly upregulated in the cancer tissue. So we found more than 3,000. And what we found very interesting and kind of nice, it was the fact that the most enriched disease-related group was lung cancer. So peptides that map to lung cancer-associated proteins, so proteins that are known to be overexpressed in lung cancer, have more peptides that are upregulated in lung cancer. So this makes biologically a lot of sense, and these peptides are here mapped in blue, so it's a lot of them upregulated, so it makes a lot of sense. We repeated the same for class 2, and the picture is very similar. However, we have a bit more downregulated peptides than upregulated. This is possibly because these tumors are rather cold tumors, and so you then have less immune cells in the cancer tissue than in the healthy, and this then will lead to a downregulation of the class 2 immunopeptides. What I would like to focus on is the protein set. Here, the peptides mapping to this protein are depicted in green, and we have only 2 that are downregulated and a number that are upregulated. Again, showing the complexity of the immunopeptidome, because it doesn't, if one protein is overexpressed, it doesn't mean that all its peptide will be overexpressed on the HLA because of the different processing machinery that is required as well. And I would like to focus on this one peptide. This one peptide, it's one of the most upregulated as well as significant. And so I want to walk you through the raw data for this one peptide. I will show you both MS1 and MS2 XICs. So we have for the healthy one here in white, and for the cancer tissue matched here in red. We have at the top MS2 XIC, so this is the different fragments. And at the bottom, the MS1 XIC, so this is the different isotopes. And what we see is no signal in the healthy and a very nice signal in the cancer, matched cancer tissue. And we can do that also in individual 10, again, very similar picture, or in individual 11. Here, the difference is much, much smaller, but we can do that across the entire cohort. And what we see is that a large majority of the cancer tissues have a very strong upregulation of this one peptide. So yes, we can find expected tumor-associated antigens. We also looked in other ones, and these also for both classes. Next, I want to present to you a collaborative project that we did with Divumet, where we were looking for neoantigens. Here, you need a little bit of a different pipeline. So the backbone is the same. However, the same sample were also whole exome sequencing. We then did variant calling and translated that into a FASTA file, so with protein sequences that we could then use for our data analysis. This was a cohort of colorectal cancer. So it was 20 samples of colorectal cancer, each with 15 milligrams, fresh frozen. We performed class 1 and followed by class 2. Overall, again, about 100,000 peptides identified across the 2 classes. But what was already very interesting, that in this case, on average, we found more class 2 than class 1. This is likely due to the fact that these samples were more hot in the sense that they had more infiltration by the immune system. Again, we can look at the different samples, and we reproduce the less biological variability in class 1 than class 2. And overall, the fact that for a number of these samples, we have really a large amount of class 2 immunopeptides. Could we find neoantigens? Yes. For class 1, we found 11, mostly coming from point mutations. For class 2, 14, again, mostly from point mutations. And overall, we had 15 cancers that had a high mutational burden. so where we expected a large amount of mutations and found a large amount of mutations. And for 8 of these, we could find at least one expected neoantigen. And now I would like to show you 2 of these examples of these neoantigens. The first one maps to the elongation factor 1 alpha 1 protein. So this is already a well-known cancer protein. We found peptides mapping to this protein in all the samples we analyzed, but the neoantigen we found only in the expected sample. So if we look at the raw data for the wild type peptide that maps to the mutation site, it's a 16-mer that binds to class 2 HLA. We find this wild type peptide in 6 samples. If we look at the MS2XIC, this is the upper part here, we see a very nice coelution of the different fragments. At the bottom, we have the MS1 isotope envelope, again, very nice isotope envelope. And then we have the fragmentation spectrum here, where we have in blue the B ion fragmentation series, and in red, the Y ions. So really a nice spectrum. And in the one sample where we have the mutation from alanine to threonine, we find this peptide. So it's again the 16-mer, but instead of having an alanine, it has a threonine. And this is reflected by the MS1 isotope envelope that it's very similar to the wild type one, but is shifted by 15 m/z that corresponds to the mass change that you would expect from this alanine to threonine change. The fragmentation pattern is very similar, and the MS2XIC is also very nice and has very similar parameters as for the wild type. And this is expected because this alanine to threonine change is a minor change, chemically speaking, and you would not expect the properties in the mass spec to change dramatically. The second example is about E3 ubiquitin protein ligase, TRP12. The wild type protein is very long and it's cancer related. We find abundant immunopeptides mapping to this protein in 13 of the 20 samples. So already here we have some variability. We have a frame shift mutation that is expected in 2 samples that leads to a much shorter version of this protein with a small sequence, new sequence as well. We find the neoantigen only in 1 of the 2 expected samples. This might be explained, for example, by HLA typing or by other reasons, for example, cancer evasion or something like this. In that 1 sample, we find 3 peptides. So different peptides, one of which is even in 2 different charges, and also 1 mapping to the wild type. So giving us a very high confidence that we can, based on the immunopeptide, find this frame shift mutation in that 1 sample. So can we use the pipeline for neoantigen detection? Yes, we see expected neoantigens for both classes. So to summarize my talk, we developed a semi-automated immunopeptidomics workflow where the use of magnetic beads was key. The immunopeptides that we get are of excellent quality and high reproducibility. We get very high numbers of identification from relatively low input material. We can generate biological insights, as I showed you in the case of lung cancer. And we can find evidence of neoantigens starting from that little material. And this was with the case of colorectal cancer in collaboration with the Differentially. At this point, I would like to thank you, and I'm looking forward to the discussion.
Thank you very much, Marco, for the presentation. It was really insightful. So let's see with the questions. We have one. Thank you, Marco. That was a great talk. I have 2 questions. First one, are you using molecular weight cutoff filters for separating peptides from MHC proteins? Do you want to maybe tackle that first?
Yeah, no, we don't, because we find that to lead to a lot of losses. So we use but we find a cleaner way also because of the mass spec, so we clean it up, so to say.
Okay. And the second question is, are you processing your DAI data using direct DAI modes? How is the data processing time using unspecific digestion?
Yeah, that's a great question. We find direct DIA to perform quite well with immunopeptidomics data. However, given the physical chemical properties of these peptides and direct DIA have developed for tryptic peptides, it is essential to also have a library because direct DIA has a strong tryptic bias at this point. But we are looking into ways of improving that as well. But yeah, so it really depends on how much material you have. Direct DIA works really well. But yeah, if you really want to be that sensitive, a good library, it's still the best way to go.
And can I maybe also ask the importance of the actual then database to build your library? Because in the case of actual patient samples, they will each have kind of individualized custom database in a way because of the mutations they might have. Is that something that you have to build? Yeah. Yes,
that's something you have to build. And what we do is generate a library from all the samples so that you don't need more material. So we take a small aliquot from each sample and then do fractionation of those samples to generate a library that we then can match to the different searches. I forgot, there was also the question how long it takes to do the search. That's actually a great question. And we find that you have to try to, depending on the computer you have, you really want to try to limit the search space. So what you can do is once you notice that you have good quality data, you don't have to look for 25 MERS if you know you don't have any in them. So you can limit your search to the sphere of peptides you are actually interested in. And that will speed up your analysis a lot. Another setting that helps a lot, if there is this maximum number of PTMs that you allow per peptide, already reducing that, especially if you have short peptide, makes a lot of sense, will already reduce a lot your search space. So it's a resource management.
Okay, that makes sense. Another question is, how are you separating the peptides from the HLA protein molecules? I think this is related to the previous question.
Yes, so we do a cleanup and then separate them based on hydrophobicity.
Okay. Karina is asking, great talk, first of all. Can you explain the experimental interaction between the beads, PAS, and the antibody HLA molecules?
Once again, so she's asking about which kind of interaction, or?
Yeah, between the beads, PAS, and the antibody/HLA molecules.
So the magnetic beads have protein A, so that binds to the antibody constant region. And then the antibody, it recognizes the HLA molecules. And this is again an antibody that it's raised to recognize conserved regions so that you can target multiple HLA molecules at the same time, allotypes at the same time.
Okay. I suppose maybe a follow-on question to that is that would you use a combination of antibodies to get a broader coverage, especially with the MHC2 peptides, which seem to be more heterogeneous?
Yeah, we never tested that. Also in the literature, for class 1, there is a very clear preference for one specific antibody. Yeah. And that works really well and recognizes all HLA class 1, the major, the canonical tree, so A, B, and C. For class 2, it's a bit more challenging. And so that is something always that you have to look exactly which one you want to pull down. And likely it would be good to have different antibodies.
So it could be very study specific.
Yeah. Yes. Yeah.
Okay. Next question is, how do you prepare a library for clinical samples when you only have a small needle biopsy to start with?
Yeah, this is usually, we don't perform a single sample analysis. So it's always within a study. So you can generate a library that's specific to the study. So you don't need extra sample. You just take a little bit from each sample and pull that. So that's our approach.
Okay. Then we have a question about, I think again, about separating. Do you mind explaining the column that you use for the separation of HLS peptides?
That's proprietary information. So we did a lot of tests in a lot of these steps, and we have some, so to say, not tricks, but you need to test a number of things and optimize that. So I will not say exactly how we do that, but it's a cleanup, so to say.
Okay. How do you determine the neoantigens come from a specific protein?
Yes, that's a great question. It comes with RNA-seq data. So with RNA-seq data, you need to do mutational calling. So that's the part that then defines whether or not it comes from there. And then you really want to make sure that at the proteomics level, that the peptide doesn't match to anything else. So we use a very, very large database with all different isoforms, wild type calling as well, to really be sure that you don't have that peptide that matches to something else.
Probably very good quality spectrum and match also MS1,
and that,
MS2.
Of course, that
to make
definitely
sure
as well.
It is right. Okay, there was one question about what is actually true discovery. I actually had this into one of your slides.
It's our approach. So it's kind of the combination of all these steps and the fact that how exactly we do mass spectrometry. And that's kind of our proprietary way of doing mass spectrometry. And that's through discovery.
Okay. So I don't see any. Here's one more question that is popped up. Really interesting. Thank you. What are the main advantages of this method compared to the genomics-based approaches?
That's a great question. It's mostly about prediction, because with the genomics, you would want to predict, based on the genomics data, you want to predict the neoantigens that are expressed and immunogenic. We try with this approach to kind of cut and make the prediction a bit easier, because you actually measure the things that are actually expressed by cells. And so the likelihood, if you have a neoantigen that is expressed, it's also much more likely to then be immunogenic. While based on genomics, you do not know if that is expressed. So you are kind of increasing your chances of success.
Yeah. Makes perfect sense. Okay, I think this is all the questions I have for now. I'd like to thank all of you for coming on board and joining us today. And of course, to you, Marco. It was really awesome to hear about your work, and I really enjoyed the presentation. And see you guys all for the next episode. Thank you.
Of course. Thanks a lot. Bye-bye.
Bye-bye.